Good afternoon, everybody. I'm Dr. Anitha, working at MES College, Bangalore, Karnataka, India. So, the title of my presentation is AFLP marker for estimating relatedness and variations among normal tissue cultured and AMF treated plants of Andrographis paniculata. So these are the plants, the vegetative shoots and the reproductive shoots of Andrographis paniculata. Andrographis paniculata is one of the highly potential medicinal plant used all over the globe, and this plant has been a main native plant of Sri Lanka but distributed in various parts of the world and cultivated in few parts of the world like China. So China, they use this plant as traditional Chinese medicine in TCM, and not only in TCM, in different systems of Ayurveda, Unani and different Siddha medicine in pharmaceuticals and therapeutic industries. So Andrographis paniculata is usually commonly called as king of bitters. That is because it is not similar to neem, saying that it is bitter. Neem as a director is also a bitter plant, but Andrographis paniculata is more bitter than neem. That's why it's called as king of bitters, and usually in some parts of the world it's called as creat, C-R-E-A-T, and in Sanskrit it is called as kalmek. And this is a highly medicinal plant, which is a potential plant which is of commercial importance in pharmaceuticals and therapeutic industries used all over the globe. And this plant, this is very potential because the plant contains certain secondary metabolites or the active principles called as andrographis. The main constituent is andrographis. That is why many pharmacological properties. In addition to andrographis, they also contain the oxoandrographis, didehydroandrographis, deoxyandrographis, neoandrographis, etc. So this is the molecular structure of andrographis structure. It contains esters of lactones. And these are the pharmacological properties. There are many properties of Andrographis paniculata, but of them, these are very important. And one of the most important one is they are trying in vitro in Andrographis paniculata to kill cancer cells, and also used in HIV, and it's a very important hepatoprotective drug. Hepatoprotective, that is protecting the liver tone, like improving the liver tone muscles of the liver. And in addition to this, hypoglycemic and also an immune system enhancer, that is why that is used in H1N1 also. The pharmacological active principle is also used against H1N1, which was prevalent recently. So the main objectives of this study are micropropagation, as already been told by many of my colleagues and my mentor. And the next one is utilization of AM fungi for better growth and performance. Elite clones are screened, and later we are subject to phytochemical analysis of primary metabolites and secondary metabolites. So the primary metabolites includes the starch, carbohydrates, amino acids, proteins, etc. Chlorophyll also we have estimated. Along with that, the secondary metabolites, andrographis. So here I have just shown andrographis. I have done primary metabolites, but our interest is on the content of andrographis. So next is germplasm conservation, and to evaluate, one of the additional topics I have taken in my research is using DNA fingerprinting along with micropropagation and mycorrhization. So to evaluate genetic diversity by using DNA fingerprinting technique by using amplified fragment length polymorphic markers. So these are the materials and methods. So the quaternary nodal segments were taken for my experimental studies in addition to different explants, and the quaternary nodal explants were like better in both micropropagation and they showed improved results. So the customary tissue culture techniques were carried out and the customary microization procedures were also carried out by using two species, Glomus marsae and Glomus fasciculatum, and the different formulas are tabulated. And these are the different morphological parameters taken into consideration in my experimental studies. The analysis of andrographis was done by using HPLC, high performance liquid chromatography, by using the mobile phase acetonitrate water and phosphate buffer 65:35. The materials and methods of the genetic diversity were also proceeded along with micropropagation and microization using AFLP markers, which is a very tedious one and an improved technique over RAPD. That is rapid, that amplified fragment polymorphism is more rapid and conventional than RAPD. That's why we could select this, and it is like the results got in one gel is almost the same for all the gels throughout. But RAPD, the results got at one particular gel may not correlate. That is why we took up to AFLP. It is very concise. The molecular evaluation and comparison of different samples were taken by using the micropropagated plants, by using control plants, and the inoculated with Glomus marsae and inoculated with Glomus fasciculatum. So the isolation of the DNA was done in the leaves of all the samples, and the leaves were very young and the plants were just 1 month old, and 0.5 grams was taken to standardize DNA for all the samples. And after standardizing, the purity assessment was done, and that fingerprinting was done in 4 steps. So these are the samples as already depicted. So the selection of the suitable plant material that included the leaves, isolation of genomic DNA by modified CTAB method, and DNA purity assessment was done after the isolation. And AFLP analysis includes 4 steps. That is, you find that the digestion is done by restriction, by using of restriction enzyme nucleoses, and you find ligation, pre-amplification and selective amplification. Then clustering analysis was done by using binary matrix, that is by cancelling the absence as 0, the presence as 1, and a dendrogram was constructed by organizing the clustering analysis by the NTS, that is numerical taxonomic systematics personal system, that is version 2. So these are the primers selected for AFLP fingerprinting. The 15 primers were selected. 15 primers were selected using E. coli plus MSC and PST plus MSC combinations were selected. So these are my observations. So the cotyledonary cultures, as I told you, they had the improved growth. That is why we could select this explant. In addition to all other explants, they were showing positive results. So the cotyledonary cultures, the best response of the direct organogenesis was in BAP. Next was in BAP, where there was more number of plants derived in direct organogenesis, they also showed in vitro flowering. The indirect organogenesis was best in MS plus IBA plus BAP plus AS, that is adenine sulfate, one of the cytokinins. Later, after the shoots were derived by direct organogenesis and indirect organogenesis, they were subjected to rooting. The rooting was usually done in IBA, subjected with GA3 for elongation. And later, all these rooting, after subjecting it to rooting, they were hardened and acclimatized. And 60 to 70% of survival rates could be observed. These are the vesicles and arbuscules, as already shown in all the previous examples by my mentor and all my lab colleagues. These are the morphological parameters where we took into consideration all the morphological parameters were done in normal samples and the treated samples. So the above photo shows normal after 3 months and the regenerated after 3 months. That is normal normal glomus massae, normal glomus fasciculatum, the below one, the regenerated control, the regenerated glomus massae and the regenerated glomus fasciculatum. So there we could observe more biomass. The positive results were more improved and the growth patterns were improved in the sample of regenerated treated with glomus fasciculatum, as you all can see. So these are the root samples. The above photo again correlates with the normal one. The below one correlates with the regenerated samples. So the regenerated sample, again you can see there is abundance growth of the root with its branches, which helps in the uptake of nutrients. So this is the graph showing the content of andrographis in all the 6 samples. So the last one, that is regenerated with glomus fasciculatum, showed more of andrographis. So there is a positive correlation between the morphological parameters and the isolation of active principle, that is andrographis. So this is DNA fingerprinting done in all the 6 samples, and after we could isolate the DNA by using agarose gel. And this is the purity assessment done after we isolated the DNA. The AFLP profile. So this AFLP profile with all the steps, which included the restriction, ligation with adapters and pre-amplification and selective amplification. After that, we usually take up the page, that is polyacrylamide gel electrophoresis, and this is the AFLP profile, and this is a profile of normal and regenerated, wherein you can see the arrow marks representing the polymorphic markers where they are not resembling, there are many polymorphic markers. So the AFLP profile of normal glomus massae, normal glomus fasciculatum on the left side, the right side showed the fragments containing the regenerated samples of glomus massae and glomus fasciculatum. And this is the table depicting the complete tabular column of the number of markers, percentage, and how many polymorphic bands were present, etc. So of all the samples, the regenerated samples were better even in the polymorphic levels. There was so much of variation in the regenerated samples along with regenerated samples treated with glomus fasciculatum. The percentage of the regenerated and glomus fasciculatum ranged from 78 to 79. This is the dendrogram constructed after the construction of a binary matrix. The dendrogram was constructed by using numerical taxonomic systematic personal system version 2. So this again shows there are 3 clusters, as you can see. The normal is correlating with NGF, regenerated correlating almost similar to RGF, that is regenerated with glomus fasciculatum, and regenerated glomus massae and normal glomus massae almost related to each other. So to conclude, Andrographis paniculata being highly potential medicinal plant, conservation is needed on this particular this day, and it is apt to conserve the germplasm, and there was a positive correlation in all the present studies, morphological correlation, there was correlation with chemical, that is active principle, and also molecular evaluation. So this is my conclusion. My acknowledgements goes to my supervisor and mentor, Dr. D.H. Tejavati, and I also thank the organizing committee of this prestigious conference for giving me an opportunity to come to China and present my paper. Thank you. Any clarification? So thank you.